Hello, folks. I'm Katie.
I'm Vinny, and this is Learn Real Good.
Wow.
Season 3.
Season 3, Episode 1.
Boom.
This is exciting.
I'm very excited.
It's been a while. How many months since we last recorded?
Since we last recorded?
Yes.
I'll say at least one as a minimum.
Do you really think we recorded a month ago?
No, just at least 1 month ago. It's
Okay.
probably more.
Very Price is Right of you.
Yeah, thank you. I want to hedge my bets.
1 minute. I'll wager 1 minute.
You're really going low.
I think it was in months, 5 months ago.
That's a good rhythm.
Yeah. It's been a while. But we're back. We're so happy to be back.
I love doing this podcast.
10 new guests.
10 new topics.
20 new facts.
30 new months
Whoa.
in the future.
Hey, well, there hopefully will be.
To be D.
Yes, let's not double down
No
too much.
promises.
No promises.
No promises.
There's an asterisk beside 30 months.
Correct.
30 or more, maybe.
At least one more month. That's all I can guarantee.
One day. I'll wager a day.
Oh, dang. Price is right to me again.
Well, it's been a while, so what's new? What are some things you want to share? What's new in Vinny Land you want to share with our listeners, the learners?
I'm looking forward to just biking more.
Ooh,
I've
yeah.
really gotten into biking around. Like, I have a car, I have kids, we drive around. It's useful.
We don't let the children drive.
Not yet, although the oldest is about to start.
Yeah, it's true. It's true.
So that's coming. But I'm trying to trade in car trips for bike trips wherever possible. And like, 1, the price of gas is motivating.
Oh, yes.
2, the planet being destroyed by me, motivating to not do that. Just like good exercise. It's all good biking around. And in this city, you can take your bike outside of rush hour on the metro system. So you can really go pretty far.
Now, I'm going to admit something embarrassing to our learners
I love
because,
it.
vulnerability, I think, is
important. It's key.
underused, and it'll bring us closer. I love biking. Every time I get on my bike the first time, I'm like, this is awesome. It's like riding a roller coaster. I feel a childlike glee. And yet, as you know,
yes,
I have not ridden my bike yet.
No, because
I'm scared. Every new year.
Yeah, every year you're like, I don't think I want to do this.
Well, I don't remember. I feel like, am I going to forget how to do it? I'm so klutzy. I'll get hit by a car.
Right,
I don't know.
sure.
Just everything about it. I'm afraid to see how much biking fitness conditioning I've lost.
Sure.
Can I do that big hill outside our house?
The big
I don't
hill.
know.
It's intimidating, that hill.
And so I keep putting it off. I find excuses to not bike. But I know I love it. And it's like, why
every
can't
year
I
you
just...
do this too.
Every year.
Yeah.
It's tradition.
It's a tradition for you to be terrified of starting something. And then as soon as you do, you're like, oh, I love this.
Why didn't I do it sooner?
Yeah, correct.
So, yeah, but I won't learn.
That's okay.
I won't learn that lesson.
No one's asking you
I'm
to.
going to keep doing this.
That's not what this show's about.
No, this show's not about learning.
No.
Well, hopefully it will be about science.
Science.
Less about
You're great at
ourselves.
learning about science.
Love it.
Improving yourself? Less so.
At least I'm aware. At least I'm self-aware.
That's the first
Self-improvement's
step.
next.
Oh, being aware of areas of improvement.
Boom.
First step to improvement.
Yes.
You can stop right there.
I
Being aware.
have, but I'm trying to get to that next step. But yeah, how the show works. We shoot the breeze. Check. And then we're going to move on to science facts, where Vinny and I have researched a science fact that we're going to share with each other.
Yes.
And you. And then we're going to get to our guest. And we do have a fantastic guest,
Super
a fellow learner,
That's right.
a Learn Real Good fan,
I love it.
and scientist. And I cannot wait.
Someone who reached out to us.
Yes, as you can too. At
Dear
the end
listener.
of the show, we'll give you the deets. But for now, fact time.
Science facts. Facts.
We came in with different theme songs.
What was yours?
Fact time.
And I said facts.
Science facts.
That's the
Yours is
recap.
better. I think-
You can rewind and hear that again if you want.
Yeah, I think you've done that before, so I think yours is the official one.
We can come up with 1,000,000 of them. Science facts. It's time for facts. Facts for science.
Okay, I'm gonna mix that with some
Yeah.
dance music, and that will be saved for the future. Okay, I'll just layer those two things.
Science facts. Science facts. Here they come.
As far as I know, the only sugar Vinny's had is the honey in his tea, but he is ready. Are you ready to share?
Yeah, do you want your science fact?
Oh, yes, give it to me.
It's hot and fresh out the kitchen,
I
coming
would
to
love
you
a
live.
nice steamy bun of science. I would love a bun right
I
now.
would love a Stevie bun.
That would be lovely.
Why isn't that an option at dim sum?
Can I order your science fact? at
Yes, you
dim
can.
sum?
All right. Well, science fact. Did you know, Katie, that the Large Hadron Collider exists in CERN?
I did because we had a guest,
That's
Ben
right.
Davis Purcell, who works there.
Correct.
Yes.
Well, did you know it was shut down? Yes.
Did he ram his car into it like I suggested
No,
he might
Ben
have?
didn't do anything accidental. What happened was it was a scheduled shutdown.
Oh.
So the accelerator, a famous accelerator in Switzerland and on a little bit over the border in France as well. It's a big circle. It accelerates particles. They smash them into each other. They have detectors looking at what comes out of those collisions.
Yes.
And it was humming along very nicely for several years.
Yeah.
But they planned a shutdown to do upgrades.
Okay.
And so what they did was they replaced some of the pieces. They upgraded it all along the accelerator, and now it can do even more collisions and different kinds of collisions.
Okay.
And so it's part of an upgrade path. So when you build this giant multi-billion-dollar facility, you don't want to just run it for a couple years and then shut it down. So what they have is a plan, a forward-looking plan for like the next 2, 3, 5, 10 years of what they want to do. And so this was one of the planned shutdowns to upgrade the facilities. And so now they've added a couple more experiments to detect. One is to detect protons colliding with helium, which is really 2 protons.
Right.
But that will help simulate early universe conditions.
Ooh.
And to see, I believe that's also to see antimatter that can come out, because that's a really great way to generate antimatter. It's fascinating that we can generate matter and antimatter. Sounds like a science fiction idea, but there's a proton and an anti-proton. When 2 things smash into each other, there's like different possibilities of stuff that can come out of it. And so they've upgraded to get different stuff to come out. And there's another one planned down the road to increase the luminosity, which just means the number of collisions and particles in the beam that can happen.
And so they knew that these things could be optimized while simultaneously knowing that now we can't do it, but one day surely we'll be able to do it.
Well, it's just because like you don't want to. You have a final state of the experiment, but it costs a lot of money to get there. So you just do like, all right, we'll do this for 3 years, and then we'll have the money saved up to do the next upgrade, and then you upgrade it, and then you run that for several years. So this is going to run for about 3 or 4 years, and then they'll shut it down again, upgrade, and then run again for 3 or 4 years.
Oh, so it was a money thing, not a technological
No, it's not like
thing?
they can't do it. It's just a question of like, all right, well, let's run it and improve it step by step over several years.
And it's the only one.
It's the biggest one in the world. The whole world has pooled all their money to make this thing happen.
Are there other?
There are others that are much smaller, but you're never going to beat like this international consortium. In theory, it's run by the EU, right?
Yeah.
So all the member states contribute to it, but there's also non-member contributors, including the United States. So the United States was supposed to build their own CERN.
Of course.
They actually dug the tunnel out and then decided not to go ahead, and they buried it.
Stop it.
No, that's a whole other story.
Where?
In Texas. It's a fascinating story. Oh, maybe I'll save that for another science fact.
So they couldn't even find another use for the giant hole they already paid to build.
I'm going to save this story.
Okay. Well, I'm curious.
Yeah.
Let's go find it and dig it out again.
No.
Make it a pool.
We can't do that.
What a waste of a hole.
Hey, man. All right, Katie.
Okay. Now, for my science fact, Vin, we are talking giraffes. When you think of a giraffe, what do you think of, Vinny?
Really tall.
Yeah. Well, what's most of the
And
height
a funny
attributable
tongue. They
to?
have funny tongues.
They lick their ice cream cones the way you lick a ice cream
Every time
cone.
I get an ice cream cone, you think
It
you
is
say the same
bizarre.
thing.
'Cause I think I've seen... I don't know for sure now if that is what a giraffe does with its tongue, but it is a wild thing to witness.
Sure.
I just lick it like an idiot. Like, just the only way I know how to lick.
Right.
Tongue in front, down to up.
Sure.
That's all I've got.
Great. It's a good move.
But you've got a whole, like, spiral...
You're giving away my secrets.
Sorry. I feel like I shouldn't watch it. I feel like it's a personal moment. Continue in the ice cream.
I should sell tickets. Is that what you're saying?
I mean, on your OnlyFans. But, so yeah, giraffes, long necks.
Yes.
I'm surely you know that giraffes didn't always have long neck.
Well, they start off as babies.
I meant ancestor-wise.
Ancestor-wise.
Right. So yeah, if you go back a few million years.
What if they started off as eels and grew the rest of their body?
Moving on.
Yes.
Long necks, giraffes.
Yes, giraffes.
But if you go back to their ancestors, you can find giraffes with not long necks.
Deer.
We've known that.
Deer.
No. Things that look like giraffes, Vinny, but with not long necks.
Okapi.
Anyway, and so like over time, the neck got longer and longer. And what do you think is the most obvious advantage to having a long neck for an herbivorous?
They can see over cool fences.
They're eating. They have a higher
leaves.
flexibility in terms of the food they're eating, right? They
Right.
can take advantage of those higher leaves. Okay. Now, so ancient giraffes, short necks, something else going on with these guys. I'm going to show you this picture, this recreation of what people think they look like. So this is the new discovery. The tops of their heads are keratinized. So they look like they're wearing a little bike helmet. I'm going to show Vinny the picture.
Oh, yeah. Okay. They look like they're wearing bowler caps.
Yes. They look quite dignified.
Yeah, they're like little English businessmen.
Yeah, they're pretty ridiculous. Now, that picture is quite suggestive in terms of what these were used for. What do you think having a keratinized top of your head would be used
now, what
for?
does keratinized mean?
Oh, sorry. Good point. Keratin is a structural protein. It makes our hair and our nails.
Okay.
And things like horns typically
so they have
are
like
made
a
of
tough
keratin.
outer, like head shell, basically.
Yeah, they have a head shell, like a built-in
for
helmet.
headbutting.
Yes.
Okay.
For competing for mates primarily,
Right.
right? Okay, so that's what they had. And so for this ancestor, they had these keratinized skulls for bashing each other in the heads, competing for mates. But modern giraffes don't.
No, they don't.
Well, so do you know how...
They smack their necks into each other's bodies. They get a lot of velocity on that.
So the long neck is sort of double duty.
Right.
Said duty. Because it is both useful in terms of fighting, because they can slam their necks together to compete for limited mates.
Yeah.
And, which this is what the keratinized part of the skull doesn't do, helps them get food.
Nice. I know one thing that giraffes do. I'm pretty sure it's giraffes. They, when a male will go to a female, smack her in the bladder and then sniff her pee to see if she's ovulating.
You know,
These are my giraffe facts.
humans aren't perfect, but God, being any other animal would have been hell.
sounds miserable.
Anyways, yeah, so they're thinking that the neck, perhaps the driving force for the longer neck was to get food,
but also sexual
but it
competition.
also served, perhaps surprisingly, that double duty,
As a
or they
weapon.
made use of it. And then you don't have to also have a big
Keratinized
heavy
head.
skull cap, right? Because
Cool hat.
everything you build costs energy, and energy is finite.
Yeah.
So if you can have one thing does both, that is advantageous.
Double duty.
Double duty.
Giraffe facts.
Well, that's it for facts, Vin.
That was pretty cool.
It's on for the main part of the show. Are you ready?
Okay, I'm ready now.
Okay, get on your interviewer hat. It looks a lot like the giraffe crap.
That's my bowler hat. Yeah.
We have a guest today, of course, and I have the pleasure of introducing our first guest of season 3. Our guest today is Claire Edrington. She is a PhD candidate in biology at McGill University. For her PhD work, she's trying to understand how cells build and rebuild their internal scaffolding, known as the cytoskeleton. Claire is also particularly interested in PhD career outcomes, knitting, and perfecting her homemade bread and yogurt. Please join me in welcoming Claire to the show. Welcome, Claire.
Hi, Claire.
Hi.
Claire is coming to us from the brand new sound studio at McGill University.
Oh, yeah. We're very lucky in the biology department to not have had parties in a while and thus have the budget for a sound studio.
Nice.
It looks awesome.
Yeah, you sound great.
Yeah, I can't wait to hear all the future podcasts coming out of there.
I know, right? There's going to be a podcast boom coming from the biology students. Get ready. Get ready for some competition here in this.
Oh boy.
Oh boy.
We welcome it.
Yeah.
Well, welcome, Claire. We're so excited to talk about your research with you today.
Yeah, I'm excited too. Thanks for having me.
And you're a fan of how did you ever find our show?
Someone on Twitter, and now I feel bad that I forget who it was, posted about it. You guys tweeted, hey, do you know Canadian graduate students? We want to talk to them. And somebody shared that on my Twitter feed. And I thought, hey, I'm a Canadian grad student. And I think I'm vaguely aware of it through the Montreal improv scene too, because I'm aware of that. And you guys showed up in 2 of my circles of social media.
That's us.
Isn't that wild, eh?
It's a small world.
It is a small world in the SciComm Twitter world.
Yeah.
Well, we're glad you're here. We're glad you reached out to us. And now we get to chat to you.
Yay.
Yes. So let's start by talking about the cytoskeleton. What the hell does that mean, Claire?
Does it have anything to do at Halloween when I spot I cite a skeleton?
What you're
That's exactly it.
skeleton. Yeah, it also is the same thing as when you want to quote something a skeleton said. You cite
right.
it.
Yeah.
Yeah.
That's better.
Nice.
That was a good one.
I mean, I don't know if everybody else knows this or not, but I certainly did not when I started learning about cell biology cells actually have a skeleton. Most cells in your body have a scaffolding that like gives them their shape. Like we have a skeleton that gives us a human shape, and a giraffe has a giraffe skeleton that gives it a giraffe shape. And if you think about the different cells in your body, like a skin cell kind of looks like a pancake, and a brain cell, a neuron, kind of looks like a long filamentous hairy popsicle, hairy lollipop.
Filamentous hairy popsicle.
Yeah.
That's normally what I think
And that's
of.
because it has a hairy filamentous popsicle shaped skeleton on the inside. So like us, cells have skeletons, and cytoskeleton means the skeleton in your cells.
And what types of materials would I find? Is it all one thing, like a cell level bone, or are there different materials
It's
doing
like wood
different
and
stuff?
plastic.
Yeah.
Yeah, there are three main types that we think about. So if you think of like a building or even a skill, we'll keep the analogy at the human level. Like, so we have bones and we have tendons and muscle, I guess, different stuff. There's fibers. So cells kind of have three tiers of sizes of skeleton. So there's the actin cytoskeleton, made up of small proteins that bind kind of in a string, and they're very flexible and they branch a lot. And they have very particular uses, and they're especially useful if a cell is migrating and moving, pushing. They kind of grow in this really branched network and push the cell membrane forward. And then on the bigger side of the spectrum, you have microtubules that are the big brother, the big boy of the cytoskeleton world, because they're, instead of just being one single chain of protein, they actually form into a tube. And so it's this kind of more rigid structure, maybe more like bones as opposed to tendons. I'm losing the analogy, but so
It's working for me.
yeah. So I'm particularly interested in the microtubules, these, because it's a bit of a more complex shape. It's a more complex structure, and the dynamics of it are different. And the middle child of the cytoskeleton world are the intermediate filaments that are
Great name.
great. Yeah. They're very
name.
much like the middle child. It's kind of there's a lot of research in intermediate filament biology right now. It's for identifying what proteins make them. Are they a mix of stuff? Are they not? What exactly are they doing? But the actin researchers and the microtubule researchers are like, oh, who? The other one? Yeah, okay.
Like the third tenor there.
Yeah,
there's
exactly.
Pavarotti, there's Flamenco, and the other one.
Yeah.
It's a Seinfeld joke.
I remember that episode.
Thank you. Vinny was lost at sea with that reference. Now, Claire, when I think of, so I know very little about the cytoskeleton. Sorry to my students who might be listening. The only thing I know is when I teach it, I say basically what you just said. That's the extent of my knowledge. There's the three different materials.
Yeah.
Plant cells don't have intermediate filaments. That's the one other anecdote I've got. But one thing I've noticed is that, like, you can make a bunch of cool organelles with microtubules, right? Like centrioles are made of microtubules, and flagella and cilia, those are made of microtubules. But when I think of actin, all it's got is like muscles, which isn't terrible. But like,
It's a good gig.
it's a good gig.
Good gig. Steady work every day.
But like when
Yeah.
I think about the things we can build with microtubules, it seems to be more diverse. Is there anything about the building material that makes it better suited? Is there something about being a tube or how it's built? It's easier to build and break for it to be so dynamic that way.
Well, you're asking a microtubule researcher, so I definitely have all the microtubules are the best because reasons.
It's like the sharks and the jets here.
Yeah, exactly. We do walk around the hallways snapping at each other, pirouetting viciously. Couple things that make microtubules special. Microtubules, because they're a tube, they do have different kind of mechanical properties than actin, and they can like in a flagella in a cell, it'll be beating and waving and moving around a lot. So you need good structural rigidity and flexibility and to be able to withstand this constant motion, but then always come back to the same shape. And in other places, for example, the mitotic spindle, which was one that you did not mention, but I think is like a highlight of what it really showcases the power of the microtubule in that when a cell divides, it has to construct this whole apparatus to separate chromosomes and organelles and everything that the two daughter cells need at the end of cell division. Microtubules will build this spindly looking thing. It looks like a spindle. There's all these microtubules crisscrossing, and pull everything apart. And part of what makes microtubules especially good at doing that is that they can grow into filaments and grow and grow and grow. And actin can do that too. Actin can grow and grow and grow. But what is special about microtubules as a polymer, because they are a protein polymer, they can shrink spontaneously. So they can just grow, and then boop, they start shrinking. Where other polymers will grow as long as there's subunits around, they'll just keep getting longer and longer. But microtubules have this like ticking time bomb mechanism that allows them to grow and shrink and grow and shrink and grow and shrink, which is very useful if you're looking for chromosomes in the middle and trying to align them. And it's like, if that's not quite it, then we let go. And if that's the, okay, we got it, now we pull and now we let go. So the dynamic, we call it dynamic instability. That's the buzzword in the microtubule world is dynamic instability. Yeah, that's like the hallmark feature of microtubules.
Now you mentioned a ticking time bomb. That sparked our interest. This might be getting super technical, but could you try to explain to us how is it that they sort of spontaneously combust?
Is there like a little digital clock? And it's like ticking down.
Yeah, it's like a little referee inside that like blows away. No.
Right.
Yeah, so microtubules are made of the protein tubulin. What a
great
happy
name again.
Great.
Yeah. Like really, biologists and names really crushing it.
Fantastic. And tubulin is an enzyme, and I'll throw technical terms and then we can work our way to understanding. Tubulin is a GTPase enzyme. So that means it hydrolyzes a GTP molecule. And that just means that tubulin, once it enters a tube and becomes part of the tube, it undergoes a maturation, like it ages. So it'll go from like young, happy, I have my like pocket of energy, and then that energy is consumed. And then, so if you have a tube growing and it's got new GTP, happy new fresh tubulin at the top, each of them, after a while, they mature and they consume the energy. Once that energy is consumed, then tubulin wants to like curl away and like give up. Like any of us.
This analogy is, so the tubulin starts off young and happy, then all of its energies is spent, and it's old and sad, and it wants to curl up and die, give up.
Exactly.
Yeah, it just lays down and is like, I'm done, I'm spent.
This is like Vinny right now.
Yeah, I guess it's true. I'm old, mature tubulin. I was young and happy and full of energy.
Yeah, so all the fresh new tubulin is kind of keeping everything together, because everything at the bottom will be like wanting to curl and energy is spent. And once the rate of maturation or that rate of aging or the rate of GTP hydrolysis, once that catches up to the tip and there's no more protective new tubulin at the top, well, we call that event a catastrophe, because everything truly just peels away like banana peels.
Wow.
Oh, that's fascinating.
Yeah.
So it's kind of like a train coming down the track, and if you're not building train tracks fast enough, the train just goes off the rails.
Peels.
Exactly. Yeah.
I would love to see a sped up video of this happening. The tubulin.
Yeah.
That'd be cool.
I should have recorded this from the microscope room. I could have showed you right there.
Oh.
In a podcast, less effective, but cool for us.
We could post it.
Yeah, we could post it.
That's pretty cool.
I would love a picture.
The sound would just be the rumble of the microscope going.
Well, this is really cool. So like having these like structures grow, and then like, okay, we're out of time. Pew! Just end of story. Catastrophe.
Yeah.
And is it at sort of a fixed rate, or are there things that can make it happen faster or slower?
That is a wonderful question, Katie.
Thank you, Claire.
Yeah. There's many different things. In a cell, I think the most prominent way of controlling how fast a microtubule will grow, or how long it'll grow before it catastrophes, or how fast it shrinks after it catastrophes, all these things are controlled by, we call them MAPs, because that's an acronym, because biologists love their acronyms. They're microtubule associated proteins.
Nice.
Another good name for you, Vinny.
I wasn't going to say it, but I was going to think it.
And different MAPs will control, like, for example, like I mentioned, the mitotic spindle that will separate all the chromosomes. When a cell goes from not being in mitosis to being in mitosis, the whole cytoskeleton, the whole microtubule network has to rebuild itself, like totally break down and rebuild itself to make that spindle. And then from a spindle, then become just a network of microtubules that does other things that are not involved in mitosis. So different proteins will say, okay, time to grow, time to shrink, time to divide, time to do this, stop, go, branch, not branch. There's a lot going on.
That's fascinating.
That's crazy.
Is that happening in me right now?
Yes.
Whoa.
Well, you're not growing so much, but anytime you have to make new cells to repair, or right, you have to...
Well, in theory, I'm repairing parts of me.
Yeah.
And even a cell that's not actively dividing, its microtubules are constantly growing and shrinking and replenishing and moving things from one place to another. Yeah.
Is there any sort of relationship with the loss of ability to build microtubules and aging? Like, oh, old people are wrinkly because that's their microtubules not building properly. Am I right?
Yeah, there are definite links. In general, any problem that you have with microtubules is a no-no, is a bad problem.
I've got a lot of problems.
Yeah, I got a lot of.
I got a big problem with those guys.
Yeah, I think the biggest one we talk about a lot in our lab is. If you can't control your microtubules, you can't control cell division. If you can't control cell division, that can often lead to cancer.
Right.
That's a biggie. Another one where microtubules are particularly important is in neurodevelopment, because when your brain is developing, neurons have to go from the center of your brain to the outside and build your cortex and build all the thinky parts. And
technical term.
Yes.
Another great biology name.
I never grew a thinky.
And microtubules play a big role in cell migration and in extending the axons and the direction that they need to go and stuff like this, and controlling how many branches the neurons make. So if microtubules are poorly regulated or misregulated or lose their regulation, then you can get neurodevelopmental issues or oopsies. Another technical term.
Yeah.
And same with aging too. If you start getting some proteins that normally would make a microtubule happy, if you have too much of them, or they start to aggregate and become a big blob that isn't very useful, that can contribute to aging and Alzheimer's and these diseases that we associate with old age.
Fascinating. So I guess, Claire, the question is now, what was your research? Like, what was your role in all this? What did you look at?
Yeah.
She discovered everything she said so
all
far.
of it. It was all me.
Amazing. What a guest.
I've been around since the 50s.
So youthful.
Yes. Well, her microtubules are working in peak form.
I guess so.
No oopsies.
No oopsies so far.
I would like to just make the disclaimer that I did not discover microtubules, and that was the work of a lot of people. And
I'm changing the Wikipedia right now, Claire, on the microtubule page. Claire did it.
Yeah, Claire did. One of the things that I worked on, and one of the things I was working on today before stepping into the fancy recording room, was something that I did not tell you about yet.
Okay.
Yeah. We'll take a step back here.
Sure.
Please.
When we think of a cell, and I have a tendency to do this too, and this is how we're all taught, you have your membrane, you have your nucleus. Sometimes you'll have microtubules, sometimes not. And you have your organelles kind of floating around in the cytoplasm. When we learn about it, it's like the liquid that's inside the cell that's not organelle or that's not nucleus. And when we think about it, we often think about this clear liquid that's kind of like water, and there's like, it's kind of nothing. But actually, cytoplasm is really, 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 really crowded and really, really, really viscous. And it's not so much like water, it's more like honey with cereal in it and Lego blocks. And
delicious. Except for the Lego blocks.
Except for the Lego blocks. So when we think about, okay, how is a cell controlling its microtubules, its cytoskeleton, it's not just a question of how much tubulin am I adding onto the end of an existing microtubule. It's how do I get tubulin through the honey, through the Lego blocks, through the cereals to get there and to make this tube grow in this crowded, viscous environment. So that's what I've been thinking about most these days, is how do the physical properties of the environment that a microtubule lives in affect how the microtubules behave. I work a lot with in vitro reconstitutions.
Oh, yes.
Fancy,
Katie knows all about them.
Right? Fancy words for saying instead of looking at microtubules in a cell, I'll take tubulin and put it in a test tube and watch the microtubules form on their own without all the other stuff.
Cool.
That's very cool.
So
Do
because
they behave differently?
yeah, in fun ways, they're a little bit slower if you don't have all the extra microtubule associated proteins that are in a cell. And they behave differently because often when we'll do things in vitro or in a test tube, we just put a buffer that's basically just water and salt, which has a very, very different viscosity than cytoplasm.
Sure.
So that's one of my projects now, is seeing, okay, but if I artificially increase viscosity by adding different molecules, do microtubules grow differently? And they do.
Whoa. So I'd imagine just viscosity slows everything down, no?
Yes, it does slow everything down. And what's exciting and what was counterintuitive was that despite slowing down how fast tubulins can come on to a microtubule, how fast they're growing, despite slowing microtubule growth rates, viscosity made microtubules live longer.
Oh, interesting. So as much as it prevents or slows down the building, it also slows down the breaking.
Yeah, which is weird because when we talked about that like maturation process, the aging, the energy spending, the rate of GTP hydrolysis of tubulin is relatively constant. Just to say that the rate of energy spending of the tubulin or the rate of maturation of the microtubule is pretty constant. And the faster you grow a microtubule, you're winning the race against maturation. So you tend to live longer. But in the case of heightening viscosity, you're growing slower, but living longer. So we've like broken that relationship, and that doesn't make much sense.
But is it like slow? Are you somehow slowing the train down, even though you're building the track slower?
I don't think so. We have
not
some
a train.
Yeah.
Interesting.
No, but I'm trying to think of that. No, I think so. The maturation, I think, is happening still at the same rate based on some recent data. So I think it's more of a question of the maturation happens, or the train will get to the edge of the track, but we're okay about it.
Oh,
fascinating. Don't catastrophe in something.
No catastrophe.
It's like slow.
It just kind
enough.
of waits. It hangs out there.
Yeah, it's slow enough that it has time for a new slow tubulin to come on, and like slow and steady wins the race. We don't need to rush anything.
All right.
So if you were to imagine sort of the dream viscosity scenario.
And who hasn't?
For microtubules.
Yeah.
We all have our own dream viscosity, I think.
Like, forgetting out what else the cell needs, just from the microtubules perspective, do you think it would be the viscosity that we do find? Or do you think it would be less viscous, more viscous, taking into account it has to both build and break? Just from the overall perspective of a microtubule's wants, what would be its dream viscosity scenario?
Trying to think of
what does it have in its secret diary?
Yeah, like what does an individual microtubule want? Are microtubules always at service of other organelles?
Yeah.
I'm not sure. I think that's a downright philosophical question.
What does anybody want?
We get deep on this show.
Yeah.
I know, huh? I know. Well, one thing that was cool with this project was that one of the molecules we used to increase viscosity in our test tube was glycerol. The sticky little molecule.
Yeah.
People have been using glycerol and viscosity as a tool to purify tubulin and to keep microtubules stable for a long time without really being able to see until recently at an individual microtubule level on a microscope. Oh, we can see at a high enough glycerol concentration, at a high enough viscosity, they just don't shrink. And this is why it works really good in purification.
Now, do you know of any like genetic disorders where someone is born without microtubules?
Without microtubules?
Like, would it be possible to live without them?
That sounds difficult.
That would be a challenge. If you can do that, you get a badge.
It sounds like really important to the structure of a cell and cell division.
Yeah, and that's
that'd be tough.
As a living organism, cell division is like your first task.
Yeah,
that's true.
There are some diseases. The thing is, tubulin, there are a few different genes that code for tubulin. They code for different versions of tubulin. And there are a few diseases that are associated with mutations in one of these genes, but they are rare. And I think it is exactly that, because division is so essential to existing, that messing with the tubulin itself is so fundamental that it's
you're going to fail out the gate there.
Yeah, exactly.
Fail out the gate.
Yeah. It's like the first hurdle. No, can't clear it.
No.
Sorry.
No. Yeah.
That's a bad one.
That's a bad one.
So how did you fall in love with microtubules, Claire?
Yeah.
Or how did you
boy.
get
What was
to
the
this
meet cute?
place?
The meat cute. That's funny. How did I fall in love with microtubules? I wanted to be a vet when I was a little high school scientist. And so I did my undergrad in biochemistry because to get into vet school, you need biology credits and chemistry credits. And I thought, let's do them together. Biochemistry. Here we go.
Smart.
Yeah. In my undergrad, I took a biophysics class. I thought the single subjects are not for me. I gotta have combined words: biochemistry, biophysics.
Yeah.
Two for
Physical
one.
chemistry.
Exactly. Every time.
Bio gym.
Yeah. And in that, so biophysics was a really, really cool class. It was intro to biophysics, kind of early undergraduate. And the first lecture, the prof walked in going, all right, hello class. So this class is basically designed to blow your freaking minds.
Whoa, that's a big move.
That's a bold claim.
So, and it did. That class, we learned about viscosity and like how cells sense their physical environment and, you know, beyond genes, beyond biochemistry, beyond cell biology, how does the physical influence the biochemical and life? So that kind of led me, like learning about cell mechanics. And I worked in that prof's lab for a bit too. Learning about cell mechanics brought me to microtubules because they are like an important part of the scaffolding and are an important part of the structural mechanics of a cell. And I also really just wanted to live in Montreal. So when I was looking at grad school, I was looking through the profs at McGill and I saw, Dr. Gary Bruhard is studying microtubules. And he's in Montreal. That's a win-win for Claire. So
Katie too. Katie.
That's how I ended up here for my PhD.
Oh, yeah?
It's like, I will do anything as long as I'm living in Montreal.
Yeah. And it was very cool too, because in my undergrad, I had kind of worked in a bunch of different labs and learned a bunch of things. And so by the time I actually found this lab, when I asked my professor in my undergrad for a reference letter, and he was like, oh, Gary, yeah, I'm going to McGill tomorrow. I'm just going to talk to him in person. I'm not going to bother writing it down.
Nice.
Right, and this was the 40s, so much easier.
Yeah. The next day, we got an electric washing machine.
It was a big week for you.
Yeah.
Very cool. And so you mentioned high school you wanted to be a vet. Were you always interested in science? Like, what was little Claire into?
Was it the science or the animals?
It was the animals. Oh yeah. Oh yeah. I was riding horses and we had a dog and a cat. And I went to vet camp when I was in 8th grade.
No, hang on.
Whoa.
Yeah.
Whoa, rewind.
Whoa, wait.
Vet camp.
Oh,
Like when
yeah.
you were a child?
Yeah, like grade 8. It was
Whoa.
It was a day camp at the University of Prince Edward Island. And like I grew up in Ottawa and I don't know how I found this, but it was like a day camp. So your parents basically had to come with you because you were like 12, 13. And so my parents like came and stayed at a cottage in Prince Edward Island and would go fishing with my brother during the day while I went and dissected a fish and like administered acupuncture to an orange and watched a cow get dissected. It was a great time.
And did you so little Claire was like, this is the best camp ever?
Yeah, it was the best camp ever.
Did the school know that oranges aren't really the purview of a veterinarian?
Yeah, I think they were trying to change that mindset.
Yeah.
Veg vet. You could be a veg vet.
A vegetarian. That's what the real vegetarian.
A vegetarian.
That's wild. I wonder if there's more. This is like
Is there
blowing my
plumber
mind.
camp?
Are
Is there
Maybe.
astronomer camp? There must be
Astronomer camp. Yes. Yeah. There's a lot of cool science camps out there. I was a camp counselor for a year at the University of Ottawa. They had a science camp. It was a grand old time.
I got roped into, no pun intended for what's about to come, a skipping camp.
Wait, skipping rope?
Yeah. In elementary school, this woman came to our school who's a professional skipper and was strongly advocating for skipping. Yeah,
This was her
That's
career?
what she claimed. She was advocating it should be part of the Olympics. And my friend was really like lit up by this talk. She's like, let's do it. And I was like, oh God, okay. So I signed up because she kind of roped me into it. And guess who didn't show up to day one?
Your friend?
Correct. It was
Oh my
me.
gosh.
I think it was like, I was too old. Is how old I was. Like 11. Everyone else was like 5. I'm like, oh God, kill me. My mom made me go because she paid for it, and rightly so. And let me just say, I won the prize at the end for most
Yes.
improved.
You crushed all the
I
little
crushed
5-year-olds.
those small
You
children.
just like point at them and be like, in your face, in your face.
All to say, vet camp sounds way better.
Yeah, that's fascinating. A vet camp.
But you ended up not pursuing, or do you plan on pursuing veterinary stuff after, once you finally nail the coffin and these microtubules? Back to the orange acupuncture? Or?
It was actually in that biophysics class, and I started to do research in my undergrad. And at the time, I also remember in my English class, we had technical report writing to do. And I wrote a report about teat infections in dairy cows and how that was an issue. And I was like kind of dead set on that. And that was really cool. But then molecules were even cooler.
It's true. Molecules are even cooler.
Yeah.
And all the animals are made up of them.
Yeah.
Exactly. I think that's kind of what drew me to research and biochemistry and biophysics more than veterinary medicine, was that I always wanted to know, okay, but why? Okay, but why? Okay, but
Right.
why? And in biology, the answer why is always proteins or
Right.
a glucose or polysaccharide or it always comes down to the molecular level. And I wanted to dive way in there.
Right. Now, was very little Claire the kind of kid to just be like, why? Like, just like pestering your parents with questions?
I don't remember. I was a very shy kid. Little Claire was not asking questions. Little Claire was quietly drawing and crafting and kind of looking outside and playing with the
Were you
dog.
writing the word why on your paper thousands of times like a lunatic?
Yeah. There's a little diary after diary.
Yeah, I don't know. I remember being very, very shy, but I always really liked school and I really liked learning. So science is kind of a great way to keep doing that, is keep learning and dive deeper and deeper into the depths of microtubules.
Despair.
I was totally the same way. I was
Yeah.
very much a shy kid.
I
Very-
can't see you shy.
I was a
Can't
little
get you to
kid.
shut up now.
As a little kid, I was very, very painfully shy of other people. Like, I was happiest just on my own reading a book.
Yeah.
Like, just drop me at the library and I'd spend hours there and be like, all right, what's in this book? All right, what's this about? And I just devour everything. I'm like that still. I'm kind of at my happiest just kind of reading and learning about things.
Yeah, I got so tired of my parents telling me to speak up and enunciate that in the 5th grade I joined the theater club, because I thought these people are going to teach me how to project my voice and enunciate.
Did it work?
Well, yeah. Or I don't know. We'll see if the recording of the podcast comes out intelligible.
Well, you sound great over here.
I couldn't hear a word you said. So do you think the theater maybe is what brought you out of your shell? Because you don't seem shy now, or you hide it well.
Yeah.
But do you think that helped you in that way?
Yeah, theater helped so much. And improv. Like, theater led to improv, and these are two places where, like, it's okay to be loud. It's okay to well, maybe in theater it's not okay to say anything you want because there is a script, but like in improv, it's okay to say anything you want and to just try something, just say something. And I think that's a big part of graduate school and being a scientist, is just try it. It doesn't matter if it doesn't work. You'll try it again and we'll see what happens. Or like, just ask the silly question. It's not that silly, or it's not silly at all. Like, other people have that question too. So removing that layer of total existential fear is very helpful. Or dealing with it if not removed.
Absolutely. I think we need more of it. It's great that it sounds like your environment has been so supportive. I think a lot of other people would say that it's still scary to fail, because it's so drilled into us all throughout school that mistake equal bad, big red X, right, to be avoided. But that's not really how science works, right? You can only do great things if you take big risks, and that's inevitably going to lead to mistakes. And that, as long as you're learning, right, who cares, right? You don't have to get it right the first time.
I mean, I wish there was a magazine of null results.
Oh, yeah. There is a journal or a website or something.
There's a journal of null results.
Yeah.
Like a kind of a, not fake, but kind of
it's
fake.
like tongue in cheek, though.
Yes.
Yeah, but I think it's legit. Like, you should publish, like publishing. I mean, physics has a famous null result the Michelson-Morley
right.
experiment about the speed of light, where they thought it ran. I thought light needed a material that the universe was permeated by this ether.
Yeah.
Kind of like, oh, sound goes through air. So what does light go through? The ether. And then they did this experiment. And they're like, all right, we're going to find out about the ether. And then there's nothing. They're like, well, I guess it's just vacuum. And it was a huge famous result.
Yeah.
So that's a null result. That's important.
But also, like, if you don't publish things that didn't work, we just keep making the same mistakes. Because you look it up, you're like, oh, no one's tried this. Or not necessarily.
No, people
no
probably
one
tried it
on
or published about it.
Yeah.
Oh man, so what's next for Claire? Do you feel like research is your home? TBD?
TBD. No, I think I'm very excited about whatever comes next. I'm not entirely sure what it is. I think there are a couple elements that I want to keep and elements that I want to add. I want to keep the learning part. I really like learning new things, and I would like to learn things that aren't about microtubules. As much as I love microtubules, like what are the other things in the world? What's next? There's stuff, there's people, there's molecules, there's other things, who knows? So that part I'm excited to keep. And the thing I'm excited to add is working more in a team. As much as I have my lab mates and, in graduate school, you have your supervisor and people around you, you're constantly around people, but your project is your own and you have to decide everything.
Right.
You can get support to an extent, but like you're the boss and you're the person to choose the proper centrifuge, speed and time. And that can get really big and a lot. And my most fun that I've had in graduate school was when I had a collaborator come in and like we would do experiments together, take turns, and be like, okay, I'll do this part, you do that part. And that's super motivating. And it seems to me that out there in the non-academic world, there's a bit more of that and more team-based worlds that I'm excited to be a part of. So that might be science communication, that might be project management. I'm very open to whatever comes my way.
That's wise, man.
Yeah, for sure.
Right?
Absolutely.
As long as when you're open to things, something's gonna happen. If you have your heart set on one very particular thing, that's where we lead to heartbreak.
Flexibility and adaptability, man. That's where the
Resilience,
future's
dude.
at. Yeah, resilience, dude.
You mentioned in your bio you're interested in PhD career outcomes.
Yeah.
What does that mean?
What does that mean?
Oh man.
What does that mean?
That means when I was deciding whether or not I would do a PhD, I absolutely freaked out. So, because I thought, well, maybe I want to be a professor, maybe I don't. What if I don't? What else do PhDs do? How many PhDs become professors? Oh my goodness, panic. So I just started asking everybody I knew who had friends who had graduated, who had a PhD, who didn't have a PhD, like, oh goodness, you people out there in the world, what are you doing? And do you like it? And is it fun? And would I have fun doing that? For me, it seemed that the people I talked to who had PhDs all had jobs that I thought would be really interesting. And I thought, okay, this is something that's opening doors for me. I'm not just siloing myself into professorship or doom. So I started my PhD, but then I still had that momentum of, okay, but what are you doing? What are you doing with your PhD? And what are you doing? So with another colleague of mine, we took over organizing a monthly career seminar where we would invite somebody once a month and they would tell us about their job outside of academia. And we were focused on outside of academia because we talk to profs every day. We see them in the hallway. We know what that looks like. And we have the opportunity to ask them more because their office is next to ours. But people not in academia are not very visible to graduate students. So that's kind of been my other passion outside of microtubules is just sharing with other students what opportunities are out there. And like, somebody with a PhD in biology can be a professor, they can be a researcher, they can be a project manager in science communication, they can be in policy, they can be a data analyst, they can be, a food blogger, somebody I know from McGill, a few people. There's a lot of food bloggers out there with PhD.
For sure.
Yeah.
Why not?
Yeah. And I also worked on a McGill like McGill itself has an initiative that wanted to know where PhDs are in the world called Trace McGill. For anybody curious about PhD career outcomes from McGill University, you should check out tracemcgill.com. And that was a really cool project to be a part of part of my job there was just Googling people, see where they ended up after they graduated. Nice. And then the second half was interviewing them and recording interviews and then putting transcripts of interviews on the Trace McGill website. So that was extra cool because then I got to talk to people with PhDs that were very, very different from mine, not just biology. They had, in the English department or history or experimental medicine and like wacky things that I had never thought about. But we all kind of have very similar challenges and very similar fears, and similar assets too. A PhD tends to be recognized in a lot of places.
Sure. Yeah. Well, that sounds like an awesome initiative.
Yeah, no kidding.
I know you have that sweet sound booth. Maybe you'll make it a podcast. Just put it out there.
Science communication.
Trace McGill, hire me back. I'll do it.
This goes out to Trace McGill. Yeah. Trace McGill. Sounds like a person's name.
Hi,
Trace.
I'm Trace McGill. Undercover PI.
Well, that's been amazing. Claire, I think we're out of time.
Yeah.
Why?
We didn't even get to yogurt.
Oh.
Damn it.
Yogurt's easy.
Give us the one sentence summary of your involvement with yogurt.
Yogurt's great. You take some milk, you heat it up, you put yogurt in it, and then you wait, and then you have yogurt.
You get more yogurt.
Well, it's like a prion. You add a little bit of yogurt, it turns the rest of the milk into yogurt.
It's just, if you want to have a bacteria culture in your home, make yogurt.
Nice.
Wow.
Nice.
Yeah.
And you make your own yogurt?
I haven't made it in a long time, full disclosure, but there was several months that I was just making my own yogurt. Wasn't buying yogurt, wasn't buying
Amazing.
bread.
Some people make their own bread. You made your own yogurt.
She makes bread and yogurt.
Yeah,
you make your own bread too?
I had multiple living organisms in my fridge for a long time.
Yeast. Wow, that's amazing.
And I imagine this was during the pandemic. Was this pre-pandemic?
During the pandemic, me and all my colleagues were like, what does your yeast culture look like? How many times a day are you feeding it? Like, we all had these yeast babies that we were asking each other and like sending each other
Sounds about
pictures.
right.
Yeah.
Amazing.
Well, Claire, this has been a true delight. Best of luck in your future,
so
whatever it is. I think we all know you're going to land just fine.
Yeah.
On a bed of microtubules or actin or yogurt.
Yeah, or Tracing McGill.
Or Trace McGill. Well, thank you so much, Claire. Best of luck.
Thanks. This was fun. Bye-bye.
Awesome. Bye.
Bye.
Oh, that was amazing.
That was very cool.
Had you heard of microtubules at all before this conversation?
No, not at all. No, I've heard of tubes.
Good.
But these are smaller
Good
since they're
stuff.
microtubules.
Mm-hmm.
I also really skipped over the part where she said brains of various sources.
Yeah.
I had that little flag in my head. I'm like, I'm not going to pursue that.
Well, you only have one brain source. So to
My
find
own?
out, just joking. To find out there's multiple brain sources.
No, I know. It's like a zombie. Brain sources.
Have you ever dissected a brain?
No.
Okay.
Is that a thing that people do?
Of course. Not a huge. Well, I mean, I'm sure
some people do.
I had to, one of the colleges I worked at, we had to dissect a sheep brain.
Whoa.
Yeah.
I don't know if I would gain a lot out of it. I've seen a picture of a brain. That feels like 99% of the information I need.
I feel, you know how I feel about dissections. I didn't like doing it, but what was cool is that like brains have like 3 layers of tissue around it. So
they do?
Yeah, the meninges. That's where meningitis, the term meningitis comes from, right? To protect it.
Okay.
Anyway, peeling those off was pretty neat because it's like layers of saran wrap on the brain.
No,
TMI?
pass.
Okay. Well, Vinny's throwing up in the bathroom. Let's take this opportunity to thank our guest, Claire.
Amazing. Thank you, Claire.
Thank you, Vinny.
Thank you, Katie.
Thank you, learners.
Learners, we love you.
And why don't you send them on our socials, Vin?
If you want to learn more about Learn Real Good, check out LRG Pod. That's LRG Pod on Instagram, Facebook, and Twitter.
And
LRG
eventually
Pod.
TikTok.
And eventually TikTok. We should get a small child to show us how that works.
And for any prospective guests, what should they do?
They should send an email to learnrealgoodpodcast@gmail.com. That's learnrealgoodpodcast@gmail.com.
Thank you for listening. If you enjoy our show, please write a review. We have one beautiful review, but we'd love more.
And tell your friends. Tell all your science friends, like, hey, you like science? Listen to these dorks.
You know what? Hell, tell your science enemies.
Yeah, if you hate us, tell your science enemies. And if you like us, tell your science friends.
Thank you.
Thank you.
See you next time.
Bye.
Bye-bye.